There is probably no disagreement among biologists that the cerebral cortex is the part of the brain that most distinctively sets us apart from any other species, and that the principles governing its development may hold the key to explaining our cognitive capacity, intelligence, and creativity. For example, Gazzaniga 2008. Perhaps the most prominent feature of the cerebral cortex in all species, and particularly in primates, is its parcellation into distinct laminar, radial, and areal domains. Eccles, 1984, Mountcastle, 1997, Goldman, Rakesh, 1987, Rakesh, 1988, Shinta Gothai, 1978. Although the surface of the neocortex has expanded 1,000-fold during phylogeny, its thickness and its basic cytoarchitectonic organization appear to be changed comparatively less. However, this morphological similarity of cortical architecture and histological sections in all mammals may be misleading, since it may invite us to think in terms of a canonical cortex that only varies in size in accordance with the variations of body mass of the different mammalian species. Conversely, our current knowledge indicates that there are significant qualitative and quantitative changes in the structure of the neocortex between species, in the size of cells, in the proportion of neurons to glia, in the ratio of excitatory projection cells to inhibitory interneurons, in the appearance of new types of neurons, in the specific organization of connections, and, most importantly, in the addition of novel, highly specialized cortical areas associated with correspondingly new axonal pathways and patterns of synaptic connectivity that can certainly have a profound effect on the functional capacity of the cerebral cortex. In spite of these differences, the small size, high fertility rate, and low cost of maintenance have converted mice into an unexcelled model for experimental research on neuroscience, and particularly on basic cortical organization. Rakesh 2000. The study of the cortical development of mammals suggests that even small differences in the timing and duration of the genesis of neural cells and changes in the composition and the ratios of cell proliferation and programmed cell death in the transient embryonic zones of the developing forebrain can be responsible for the evolutionary expansion of the neocortex and the concomitant appearance of novel functions in cognitive processing of information. Bystrom, Blakemore, and Rakesh 2008. Although genetically humans are surprisingly similar to other animals, the uniqueness of the human cognitive potential, which is an output of cortical function, may have some genetic basis. Since the molecular structure and function of neurotransmitters, receptors, and ion channels do not change substantially over the phylogenetic scale, the secret to the success of Homo sapiens is probably mainly due to an increased number of neurons, more elaborated connections, functional specialization, and introduction of new cortical areas. Even the so-called essential genes, which are considered responsible for survival of the individual, give different phenotypes in different species, and about 20% of the mouse orthologs of human essential genes are non-essential in mice. Liao and Zhang 2008. It is therefore apparent that neither the genetic, the cellular, nor, most importantly, the circuitry basis of human cortical uniqueness can be deciphered by studying exclusively rodents, much as one cannot expect to understand the origin of the mushroom body of insects by studying the primate association cortex. However, for logistic, ethical, and financial reasons, the easiest approach to understand general mammalian features is to study the mouse, and the closest we can come to elucidating primate-specific cortical development with the modern molecular and cell biological methods is to analyze its development in the monkey, and specifically in Old World monkeys such as the macaque, which are much more similar to the human species than their relatives, the New World monkeys. For the preceding reasons, the present review is based on the comparative studies of neocortical development in the mouse, macaque, and human. It is limited to the early developmental events that lead to the formation of cellular constituents and their basic connectivity, the final tuning of synaptic connections and their modifications by experiences described in other communications.